Hello, my name is Benjamin Ryskeldi-Falcon. I am a group leader here at the UK Medical Research Council Laboratory of Molecular Biology, or LMB, which is in Cambridge in the UK. And the philosophy of the LMB is that we can better understand biological processes by studying the molecules that underpin them. And my group very much takes this approach to the study of neurodegenerative diseases, including motor neuron diseases and dementias such as Alzheimer's disease. I entered this field as a PhD student with Michel Goeder, also here at the LMB. So during this time I was developing cellular models of tau aggregation to study this propagation phenomenon between cells. However, at the switch between my PhD and postdoc, there were large improvements in structural biology using a technique called electron cryomicroscopy. So in this technique you can take proteins or protein complexes and freeze them very rapidly so that you preserve their structure, and you can then image them at high resolution in an electron microscope and reconstruct the 3D structures of the proteins. Now, there were advances in the imaging hardware, for example in the cameras used to take the electron micrographs, and also in the software to analyze these images, that suddenly made it possible to determine high-resolution structures. And so at the interface between my PhD and my postdoc, together with Sean Skeres here at the LMB and a postdoc called Anthony Fitzpatrick in his group, we attempted to utilize these developments in cryo-electron microscopy to determine the structure of tau filaments in Alzheimer's disease. And this was a success, and we were able to show the atomic resolution structure of these filaments. From there, we now had a technique that would allow us to study protein aggregation of tau, but also other proteins in Alzheimer's disease, but also other neurodegenerative diseases at this atomic resolution, which is what I have continued to investigate now in my independent career, with a focus on motor neuron diseases and dementias. I established my own independent laboratory in October of 2019, and our first aim was to determine the structure of TDP-43 filaments, which are the main pathology of motor neuron disease, such as amyotrophic lateral sclerosis, and of a whole number of different frontotemporal dementias. And so for TDP-43, we really didn't know what type of filaments this protein formed in disease, and we didn't know which parts of the protein formed the filaments. And of course, this information is important if we want to target the pathological assembly of this protein, either for the diagnosis or treatment of these diseases. And so, using the advances in cryo-electron microscopy that I had harnessed during my postdoc, and we were able to determine the structure of TDP-43 from amyotrophic lateral sclerosis and an associated type of frontotemporal dementia called frontotemporal lobe degeneration type B. Now, this revealed that TDP-43 forms a type of filament structure called an amyloid. This is similar to tau in Alzheimer's disease, for example. And so this really brings TDP-43 into a similar group of diseases. And it also showed us that it was a specific region, small region of the protein that forms these filaments, which now gives us the information for targeting these for diagnosis or therapeutic benefit. Now, there are many additional diseases where we see TDP-43 aggregation, for example, in additional types of frontotemporal dementia. And so subsequently, my group has focused on determining the structures of TDP-43 filaments in these additional diseases. And the large surprise from this work was that we have distinct filament structures of TDP-43 characterizing different diseases. So we have a single specific structure for amyotrophic lateral sclerosis and a different structure in different types of frontotemporal dementia. Now, this suggests that it may be possible to develop tools that bind to these filaments to selectively diagnose different diseases based on this structural information. And we're also trying to harness shared structural features between the filaments in order to develop therapeutics that are broadly useful for TDP-43. Now, an unexpected findings were additional proteins that were not known to aggregate. So because we determined these structures from the brains of individuals who had diseases, so these are donated brain samples, we are not looking for a particular hypothesis. It's an unbiased approach, and we therefore identified new proteins that form filaments in disease, such as annexin A11, which forms a heteromeric filament with TDP-43 in a specific type of frontotemporal dementia, and the protein TAF15, which characterizes another group of diseases alongside TDP-43. There are so about 50% of cases of FTD have TDP-43 filament aggregation, and about 40% have tau, and then the remaining 10% we think are characterized by this new protein TAF15 forming the filaments. So the next steps for us are firstly to use cryo-electron microscopy to determine the structures of the aggregating proteins in additional diseases. So because we find a specific protein and a specific structure per disease, we need to look at all the different neurodegenerative diseases because they will each have they're likely to have their own structure. And of course this will then inform how we target this process, either for diagnosis or therapy. So we're focusing on additional types of frontotemporal dementia with TDP-43 or TAF15 filament formation, as well as additional motor neuron diseases. Now, whilst this is ongoing in the lab, we are also trying to use the knowledge we've gained from these structures to develop the first experimental models of the aggregation of these proteins that recapitulate what we see in the human brain. So one of the findings from these structures was that the models we currently have for, say, TDP-43 aggregation or TAF15 do not recapitulate the structures that we see in human brain, and are potentially therefore not very useful for studying the mechanisms of the formation of these filaments. And so we're using the structural knowledge to develop cell models which do have the correct structures that we see in human brain. The third future direction in the lab is to use cryo-electron microscopy to image these protein filaments within the brain tissue itself. So far we extract the filaments from the human brain in order to determine their structure in isolation. But ideally we would just look at the brain tissue and at the protein in its place within the human tissue. So there have been a number of recent developments in cryo-EM that now allow us to look directly at these filaments within the human brain. So we don't expect that the structure will change, but this will give us an extra layer of information. It will show us the subcellular environment that these filaments form and accumulate in, and it may show us some of the interactions that are important for their formation and the damaging effects of having filaments in the brain. So we anticipate that this will give us a completely new layer of molecular neuropathology, which will then inform the models that we produce in the lab, and also inform strategies to target filament formation for therapy. Receiving this prize is a great honor, and there are several benefits associated with this prize that I'm particularly excited about. The first is connecting, of course, with a new community and wider community of researchers to disseminate our findings using cryo-EM and the new level of pathology that we find in these diseases. I'm hopeful that the new connections will lead to collaborations, for example, testing new models of neurodegenerative diseases where we can give structural information on the types of models that other researchers are using. This prize has also enabled me to travel to meet new collaborators and to foster new collaborations. So for example, I spent the month of December in the US visiting many different laboratories, either to study patient samples to solve new structures, and also to set up collaborations for the future objectives of the lab. And so this money has really enabled those kind of connections to be forged.